click to that one. Very few papers is only corrected. Okay. Next, I think is according to our academic timetable. Yeah, academic calendar. Our timetable is on 22nd onwards. No, not 22nd, 23rd onwards. Our second module, second mid exams, module 2. So we try to hurry up to that one. The preparedness is required. Our module 2 is water and the biomolecules, the interrelation between the water molecule. Okay, now the number is 25. Okay, the rest of people will join, we will expect to them. Today is our lab class. It's 2 hours class up to 4:50 is our class. So don't quit from this, the middle. Okay. After that, once we discuss, after that we go to the topic of bacterial growth and bacterial culture of labs. We discuss later on. That is relevant to our lab point of view. The lab session, bacterial growth and bacterial population growth is one of the topic for the lab session. Okay. In module 2, the biomolecules comes under sugar, starch, cellulose, amino acids, the proteins, lipids, nucleotides, DNA, RNA, and all this takes place. This big vital role, the water molecules, and the major elements of carbon, hydrogen, oxygen, nitrogen, phosphorus. These are all recombinate and combines. Someone is water soluble, someone is non-soluble. It's not mixed up with these hydrophilic and hydrophobic ones. And someone is condensate, condensing and elimination of water, polysaccharides, monosaccharides. All these takes place in biomolecules structures. The nucleic acid, then nucleotides, especially the nucleotides, those particularly the nucleotide arrangements and everything, and the structure and function of proteins. Proteins is major. The construction of body, even the body fluids, contents of the water, structure of the body and shape of the body, especially in the living things of animals. Yeah, the plant tissues and their structure. There it is, the flow of the water, which reach towards that all the metabolic activities, catabolism and anabolism, all these point of view. The next is our hemoglobin, antibodies, enzymes, industrial applications. Yeah, enzymes, industrial applications. A lot of bacteria is released. Their enzymes acts as some medicines. Yeah, and next is fermentation. fermentation based industry is alcohol and beverages industries. Yeah, fermentation and industrial applications. The next of this particularly, these carbohydrates is one of the biomolecules. Generally, carbon, hydrogen, oxygen is contains in carboxyl groups also. The sugar, starch, cellulose as the material, cellulose materials is the building. It's used as a fuel of the building material and the building material. Yeah, it gives the energy. This fuel means here it gives the energy, and the building material it constructs means of that the cell or nucleotide it carries. So these sugars, especially sugars, are monosaccharides, and starch and cellulose is polysaccharides. What it is monosaccharide and polysaccharides we will see in our video. Okay. Number of monosaccharides combines polysaccharides will generate this linkage of monosaccharide units. That's a prime unit. Then polysaccharides will generate. Yeah. Glucose, fructose. These two is C6H12O6. One is keto form and another one is enol form. Based on its functional group, ribose and glyceraldehyde. These are all comes under the monosaccharides. Yeah, and alpha glucoses and beta glucoses also. Alpha and beta glucoses, the difference is whatever the hydroxyl groups is all are at one side or opposite sides, either in a straight structure form or else in a ringed form, hexagonal ring form, whatever way we may write it. It is alternatively hydroxyl groups or all are in one side. Yeah, the alpha glucose. Opposite is beta glucoses. Maltose. In this maltose, this is the monosaccharide of the glucose is combines, then the maltose will generate, that is disaccharide, one or two the above of glucose combines, and the dehydrate is removing of the water molecule, then maltose will generate. Yeah. Cellulose is also polysaccharide. The number of the combinations of the glucose units is linked and alternative opposite of hydroxyl groups, then cellulose is fiber body. Cellulose material is polysaccharide will generates. Next, amino acids and proteins. This gives the strength of the body. Amino acids is monosaccharides, proteins is the number of proteins in a cell, that is polysaccharide. In this carbon, oxygen, and nitrogen. Very rare case of the sulfur also. It standard amino acids up to 20 amino acids we can identify. The amino acids according to their code wise, each and every amino acid is in a one letter code. We can identify those amino acids, that particular the letter code. In medical reports, the amino acids analysis is mentioned. Each individual amino acid, it's in one letter is significant. See, it's in S, T, N, or it's in a thyro, it's in a blue, it's in a val. All these are different. Okay, according to their categories. Those are all in hydrophobic, hydrophilic, and the polar amino acids. Total this particular is 20. It's classified in several groups. Proteins point of view, these are all bonds with one amino acid and carboxyl groups to another. Both adjacents is one to another is combines, and biggest polymerase of protein is generates. The next is lipids, is also one of the biomolecule. Here it is oxygen, carbon, oxygen, hydrogen is a common, oxygen and phosphorus also. But if in proteins is nitrogen, and here it is fats and phospholipids and steroids. These comes under the lipids. Lipids also is the binds with carbon, very much longer one. The fatty acids and the glycerols. These are all triesters. Okay, this energy storage and insulation point of view. This all lipids. It's used to the construction of the body, and a lot of lipids, hydrophilic one and hydrophobic one, can distinguish. The cholesterol also. The cholesterol is one of the fatty acid. Is in heart, lungs, and other vessel circulatory system. It may not at clot, but it's deposits, or as the body, high obesity is a huge water quantity, or else some fatty substance. Then it is comes under the cholesterol. This cholesterol contains a lot of effect to the animal cell and animal membrane. It just deposits a lot of the cholesterols. Here it is from all carbon and oxygen and other hydrogen and oxygen and other elemental substances also. Next is nucleotides, DNA, RNA, ribonucleic and dinucleic acid, deoxynucleic acids and ribonucleic acids. Only the hydroxyl group is only the differs. It contains nitrogen, phosphorus, oxygen, carbon. Next is nucleotides. The nucleotides is contains of this particular the letter coding here also. A, T, G, C, U, adenine, guanine, thymine, cytosine, and uracils. But these are also again water soluble, pyridines and purines and pyridines. Yeah. This is one aromatic ring or double aromatic rings, single aromatic ring of adenine, guanines, and thymine, cytosine, and uracil. These are all are two aromatic rings, one to another its compounds. Yeah, the nucleotides, monophosphates or polynucleotides. Polynucleotides is monomers like is also phospholipid bodies. Through it can generate one to another. It is the code wise. Yeah, mRNA, tRNA, transport RNA, messenger RNA, and all these are a lot of ribosomal RNA. All these are named transcription and translation and transport amino acids, and all these point of view, these usage of particularly for nucleotides of this particular code of adenine, uracil, guanine, thymine, cytosines. Next is function structure. This particular double helix structure, the great scientists of Watson and Crick, they identify as a ladder structure. It is spiral one to another. It binds with the both ends. Both ends it's in a, just like the ladder rod like, but it's twisted one. Hemoglobin. Next, one is our hemoglobin contains of the human body, especially here it is the exchange of oxygen and the released carbon dioxide. So all these carries. Already we discussed the hemoglobin contains of the ferrous iron, ferric iron, ferric heme, ferrous heme. These is bring to oxygen. Oxygen is it carries to ferrous heme to from ferric heme. Whenever it carries, the ferric is +3, Fe +3, ferrous is +2. Then immediately the exchange of that particular the electron it oxidizes ferrous to ferric. But at the same time, the ferric is very unstable one. Again, once it supplies the oxygen, and again it's in a converts as in a ferrous form and carries this particular carbon dioxide released by this the respiratory tract. Yeah. These all activities we can identify in the pH values. This particular oxygen is blended, blends with, then the particular the pH value of the blood is not at all change. Just like as in a buffer, it uses not as in a highest acidic tendency and not in the highest basic tendency. In that way, it is 7.2 to 7.4 pH. It contains. pH have no units, you know well. The higher level of pH value is significance of carbon dioxide, the basic tendency, the lower values, significance of more oxygen. Yeah. Acidic tendencies. Next is antibodies. Antibodies is defense mechanism, but it is in the heavy chain and the lengthy chains of this particular the antibodies will generate. Let's fight out to the infectious disease caused organisms, or else direct fight with infection part and the locations. These are all in the body's white blood cells is fight out. Next, enzyme catalysis. The reactions is catalyzed, the rate of reaction is decreases, then the reaction is faster than in general. The general reaction is very slow, then the rate of reaction is decreases, chemical kinetics. Then the first reactions, the lowering energy of its translate state. The energy is lowering, then it's easily the reaction is faster. Enzymes, industrial applications is also a lot of enzymes, amylase, cellulose, and this lipase, the lipase and penicillinase. Yeah, it is a synthetic one also. Through this bacterial released ones, and this someone is in the plant's one. Hydrolysis of the cell wall by the plant is ligrinase. The ligrinase is one of the enzyme it releases. The hormones also. You know about the hormones of, especially in the human, the males is testosterone, the females is estrogen, the adolescence characters is begins, they converts as an adult tendencies from childhood to boyhood to adolescence to adults. Then the growth of particular the structure of human body will stabilize. Up to that, the growth is day to day. It's going on. Yeah. The next is fermentation. The fermentation point of view, the citric acid, so many biopolymers and antibiotics preparations, biopolymers preparations, citric acid, this acetic acid, yeah, alcoholic fermentation by yeast, and through beverages, wines, and the bakeries industry, the lactic acid fermentation, especially curd making in household, curd way, cheese, and some type of foods also. There's so many foods is prepared through particularly of the fermentation. Yeah. All these activities we can find out in our biomolecules, the blood and enzymes, and at the same time the hormones is also, but hormones is very little bit. These are the things we can identify at here. Yeah, you see all these, what it is in a biomolecules, in what way it is functionalized. Yeah, now I will share the screen. This is all things in outlines is only I discussed in our syllabus point of view. Now you watch the video. Yes, I know that. Not yet share the screen. You are correct. 11031 Skype. Yeah, now I will share. Shall of you listen carefully. Oh, wait. Sure.
We obtain similar elements while doing an elemental analysis of different living and non-living things.
Is it visible?
For example, we find
See the
elements
sound
or
and
compounds.
visible. Both of the biomolecules point of view. This is in Siksha.
The elemental composition of living tissues can be determined by following a technique known as elemental analysis. Interestingly, we obtain similar elements while doing an elemental analysis of different living and non-living things. For example, we find that elements such as carbon, hydrogen, oxygen are common to the
carbon,
Earth's
oxygen,
crust,
hydrogen,
as well as
nitrogen,
living
sulfur,
tissue.
sodium, calcium, magnesium, silica. This
However,
all are combined.
the composition of these
Someone is
elements
in human
is much
body and
higher in a
crust.
living organism compared to the Earth's crust, as shown in this table. To ascertain the chemical composition of organic compounds in a living organism, we perform chemical analysis.
Biomolecules analysis.
For example, to find the chemical composition of a living tissue, such as a vegetable,
one of the vegetable product of carrot.
We
Carotenoids,
first grind it
xanthophylls,
in
just like
trichloroacetic acid.
chlorophylls like.
As a result, we obtain a thick slurry. Now we strain this slurry through a cheesecloth to obtain two fractions. One fraction is the filtrate or the acid-soluble pool, and the other fraction is the retentate or acid-insoluble fraction. The acid-soluble pool contains organic compounds. Once a compound has been extracted, the next stage is to separate one compound from the others
Listen carefully. So all words are very
using separation
important.
techniques such as chromatography and
The
centrifugation.
centrifuge, this chromatography technique,
Thereafter,
this
various
particular
analytical techniques
separation
are used
techniques.
to identify the molecular formula and the structure of the compound. All the carbon compounds we derive from living tissues are called biomolecules. For example, carbohydrates, lipids, amino acids, water, and nucleic acid. Biomolecules such as amino acids and sugars are also known as metabolites because they are intermediates and products of metabolism. They are classified as primary and secondary metabolites. Compounds such as hydrogen, carbon, oxygen, nitrogen, sulfur, and sodium, which are involved in normal growth, development, and reproduction, are called primary metabolites. They are found in animal tissues. On the other hand, compounds such as alkaloids, flavonoids, rubber, essential oils,
Metabolites
antibiotics,
is metabolism.
colored pigments, scents,
Yeah,
gums,
catabolism
and
and
spices
anabolism.
are
This
known
comes
as
under
secondary
metabolism.
metabolites.
One is construction
They are found in plant,
and
fungal,
destruction.
and microbial cells and perform an important ecological function. Some of them play a helpful role as antibiotics and pigments. Living beings are also composed of inorganic compounds such as sodium chloride and calcium carbonate. To analyze the constituents of inorganic compounds, we carry out the following process. We take a small amount of living tissue, such as a leaf, and weigh it. This weight is known as wet weight. Then we dry this leaf
Yeah,
to
wet
allow
weight,
the water to
dry
evaporate
weight,
to get
and
its
ash
dry
weight.
weight.
See differences.
Now, when we burn the leaf, the carbon compounds get oxidized and the leaf releases
Containing
carbon
of
dioxide and water vapor.
elements is
The remaining ash contains inorganic elements such as calcium, magnesium, and potassium. Therefore, analysis of compounds helps us identify organic and inorganic constituents in a living tissue. It also helps identify functional groups such as aldehydes, ketones, and aromatic compounds. In biology, however, functional groups are present as amino acids, fatty acids, and nucleotide bases, together known as biomolecules. Amino acids are the building blocks of all proteins. The amino group and carboxylic group are present on the same alpha carbon
amino
atom
groups
of
and
the amino
carboxylic
acid.
acids in both
Hence,
sides.
they are also known as alpha amino acids. Amino acids are substituted methanes. There are 4 substituent groups occupying the 4 valency positions. These groups are hydrogen, the carboxyl group, amino group, and a variable group also known as the R group. based on the nature of the R group. There are many amino acids. However, there are 20 standard amino acids
Yeah,
in
20
proteins.
standard amino acids already we discussed, but each and every one is in a letter.
For example,
L, V, I, P,
glycine,
M,
in which the
some
R
EF,
group is hydrogen,
all these are glycine,
alanine,
alanine,
where the R
serine.
group is a methyl group,
It signifies as in the first letter.
and serine, where the R group
20
is a
amino acids,
hydroxymethyl
all of
group.
you, important. This number is important.
The chemical and physical properties of amino acids are essentially of the amino, carboxyl, and the R functional groups. As amino acids contain both the amino group and carboxyl group, they can be acidic, such as glutamic acid, basic, as in the case of lysine, and neutral, like valine. Similarly, there are aromatic amino acids such as tyrosine, phenylalanine, and tryptophan. A unique property of amino acids is the ionizable nature of the NH2 and COOH groups. Amino acids behave as both bases and acids. In solutions at a particular pH, ions with both positive and negative charges are equal. This form is known as the zwitterionic form. Another group of biomolecules is fatty acids. Long chains of fatty acids and their derivatives lead to the formation of lipids. Lipids include a broad range of molecules such as fats, waxes, fat-soluble vitamins, and glycerides. They are generally insoluble in water. Fatty acids that form lipids have a carboxyl group attached to an R group. The R group could be a methyl, ethyl, or a higher number of CH2 group with 1 carbon to 19 carbons. For example, palmitic acid has 16 carbons, including a carboxyl carbon. Fatty acids can be saturated or without a double bond. They can be unsaturated or with one or more C double bond C. Glycerol or trihydroxypropane is a simple lipid. When fatty acids combine with glycerol, they form esters, also known as glycerides. Glycerides can be monoglycerides, diglycerides, and triglycerides. Based on their melting points, glycerides may be either fats or oils. Oils have a lower melting point and hence remain as oil in winter, while fats freeze easily at low temperatures. Lipids that contain phosphorus and a phosphorylated organic compound are known as phospholipids.
It's particularly cell membrane.
They are found in the cell membrane.
It's sort of matrix-like structure
Living
we
organisms
identified.
also have many carbon compounds with heterocyclic rings, such as adenine, guanine, and thymine. These compounds are called nucleotide bases, and they are important biomolecules. When carbon compounds with heterocyclic rings are attached to a sugar, they are called nucleosides. For example, adenosine and thymidine. On the other hand, if a phosphate group is also found esterified to a sugar, they are called nucleotides such as adenylic acid and thymidyllic acid. Nucleic acids such as DNA and RNA consist of nucleotides. They are molecules involved in heredity. Thus, biomolecules act as building blocks of life and perform vital functions in living organisms. All living organisms produce organic molecules called biomolecules. Biomolecules can be broadly divided into two types: biomicromolecules and biomacromolecules. On performing a chemical analysis, we find that biomicromolecules are chemical compounds found in the acid-soluble pool with molecular weights ranging from 18 to 800 daltons. Some examples of biomicromolecules are water, minerals, amino acids, sugars, lipids, and nucleotides. On the other hand, biomacromolecules are compounds found in the acid-insoluble fraction. They have molecular weights in the range of 10,000 daltons and above. Biomacromolecules, or compounds which come from the insoluble fraction, are proteins, polysaccharides, and nucleic acids. Interestingly, lipids, which are biomicromolecules, are also found in this fraction. The acid-soluble fraction represents the cytoplasmic composition, while the acid-insoluble fraction consists of macromolecules derived from cytoplasm and organelles. Together, these two fractions represent the complete chemical composition of living organisms. Biomacromolecules have molecular weights in the range of 10,000 daltons and above. Let's understand why lipids, whose molecular weight does not exceed 800 daltons, are a part of the acid-insoluble fraction.
Towards nucleic
Lipids have a
acids.
small molecular weight, and they are arranged in the cell membrane and in the membranes of mitochondria, chloroplast, and Golgi apparatus. During a chemical analysis, when we grind a tissue, the cell structure ruptures and various membranes break into pieces and form vesicles. These vesicles are not water-soluble because lipids are hydrophobic. So they get separated along with the acid-insoluble pool. We'll now learn about polysaccharides in the acid-insoluble fraction. Polysaccharides are long chains of sugars. They are thread-like structures consisting of different monosaccharides as building blocks. In a polysaccharide chain, for example in starch, the right end is known as the reducing end, while the left end is called the non-reducing end. Polysaccharides can be divided as homopolysaccharides or heteropolysaccharides. When all the monosaccharide units in a polysaccharide are of the same type, it is called a homopolysaccharide or simply homopolymer. Some examples are cellulose, starch, glycogen, and
Cellulose,
inulin.
starch, glycogen, and insulin.
Cellulose is a homopolymer
First
consisting of
polysaccharide
only
of
glucose
cellulose.
as a monosaccharide unit. Some examples of cellulose are a plant's cell wall, paper made from plant pulp, and cotton fibers. Starch, on the other hand, is a homopolymer of glucose which acts as the storehouse of energy in plant tissues. Starch forms helical secondary structures, and in that position, it can hold iodine molecules. Conversely, cellulose cannot hold iodine molecules as it does not contain complex helices. In animals, glycogen acts as a homopolymer formed of glucose as a monosaccharide unit, a variant of starch. Inulin is a polymer of fructose. Polysaccharides, which are formed of more than one type of monosaccharide unit, are called heteropolysaccharides or simply heteropolymers. They are complex in nature with amino sugars and chemically modified sugars as building blocks. Glucosamine and N-acetyl galactosamine are examples of heteropolymers. Another type of biomacromolecule in the acid-insoluble fraction of living tissues are nucleic acids. A nucleotide acts as a building block of nucleic acid and forms a polynucleotide chain. A nucleotide has three chemically distinct components, namely a heterocyclic compound, a monosaccharide, and phosphoric acid or phosphate. The skeletal heterocyclic ring compounds are actually nitrogenous bases such as adenine, guanine, uracil, cytosine, and thymine. There are two types of nitrogen-containing bases, namely purines and pyrimidines. In a purine, 6-member and 5-member nitrogen-containing rings are fused together. Pyrimidines, however, have only a 6-member
purines and
nitrogen-containing
pyridines.
ring.
Purines is
Therefore,
two rings.
adenine and guanine are substituted purines, while uracil, cytosine, and thymine are pyrimidines. The sugar found in nucleotides is either ribose or 2'-deoxyribose. Both these are monosaccharide pentose sugars. Therefore, nucleic acid containing deoxyribose is called deoxyribonucleic acid or DNA, while the one with ribose is called ribonucleic acid or RNA. Both DNA and RNA carry genetic information and are found in the cells of all living things. Together, polysaccharides, nucleic acids, and polypeptides or proteins form the macromolecular fraction of all living tissues. Proteins are important biomolecules needed by all living organisms. They are polymers of amino acids arranged in a linear chain and linked by peptide bonds. Therefore,
Peptide bond.
they are also known as polypeptides.
Amino acids.
Amino acids that go into the making of proteins can be essential or non-essential. Essential amino acids are found in our diet, while non-essential proteins are synthesized by our body. There are 20 types of amino acids that come together to form proteins, and so they are also known as heteropolymers. In a heteropolymer, there are different types of monomers repeating n number of times. Proteins form very interesting three-dimensional structures. The structure of molecules is depicted differently in different disciplines. In chemistry, structure means the molecular formula. For example, salt is also known as sodium chloride or NaCl, where NaCl is the molecular formula and not the structural formula. On the other hand, organic chemists present a two-dimensional view of molecular structure. For example, in benzene or naphthalene, this structure represents the structural formula, whereas the molecular formula is C6H6. Physics considers a three-dimensional view of molecular structure. However, biochemists have described the protein structure
Yeah,
at
primary,
four
secondary,
levels:
tertiary, quaternary
primary,
protein structures. How to they arrange
secondary,
in additions
tertiary,
of one
and
is nitro
quaternary
group,
structure.
another is carbon groups.
Let's learn more about them.
You see,
The primary structure is the simplest structure, which refers to the positional information of amino acids in a protein. Hypothetically, a protein is a thread formed by amino acids where the left
the
end is
amino group
represented by the
and
first amino
carboxyl
acid
group,
or the N-terminal
carboxyl
amino
group.
acid, and the right end is represented by the last amino acid or the C-terminal amino acid. The protein thread is represented as a horizontal straight line so that the position of the amino acids can be determined. However, in nature, proteins are not completely straight but are folded in the form of a helix, somewhat like a revolving staircase. In the secondary structure, amino acid links get longer, and due to the heaviness of the molecule, it tends to bend. This preliminary bending is considered to be the secondary structure. It is either an alpha helix or beta sheets. The third level of protein structure is the tertiary structure. It refers to the three-dimensional structure of the entire polypeptide chain.
Hemoglobin.
The function of a protein, except as food, depends on its tertiary structure. If this is disrupted, the protein is said to be denatured and it loses its activity. The fourth level of protein structure is the quaternary structure. This structure is formed by several protein molecules or polypeptides, also known as protein subunits. These subunits are arranged as a linear string of spheres arranged one upon each other in the form of a cube or plate. For example, adult hemoglobin consists of 4 subunits, 2 of which are identical to each other. Therefore, 2 alpha-type subunits and 2 beta-type subunits collectively form human hemoglobin. Some of the most common proteins we come across are collagen, rubisco, trypsin, insulin, antibodies, receptors, and GLUT4. Let's learn more about each of these proteins. Collagen is the most commonly occurring protein in the animal world. Collagen fiber is generally found in connective tissues and acts as an intercellular ground substance in the space between the cells. On the other hand, ribulose bisphosphate carboxylase-Oxygenase, or rubisco, is the most abundant protein in the whole of the biosphere. It acts as a catalyst during the process of carbon fixation. Trypsin acts as an enzyme, and insulin acts as a hormone. Another protein called antibody helps fight infections. Receptor, another protein, helps in sensory reception, while glucose transporter 4 or GLUT4 helps in transporting glucose to the cells. In this manner, Various proteins play a vital role for every living organism. The structure of these proteins is crucial in determining their functions. Bonding plays a crucial role in the making of a polymer. A polymer is formed when many monomers come together and bond in long chains. For example, polyethylene is made up of repetitive units of ethylene monomer. Let's look at the bonding in biomolecules such as proteins, polysaccharides, and nucleic
Biomolecules
acids.
classification.
Proteins are amino acids linked by peptide bonds. This chemical bond is formed when the carboxyl group of an amino acid reacts with the amino group of the next amino acid, losing a water molecule. This process is also known as dehydration. Polysaccharides are formed when several monosaccharides link together by a glycosidic bond. This bond is also formed due to dehydration. The glycosidic bond is a functional group that can exist between two carbohydrate molecules or between a carbohydrate and a non-carbohydrate molecule, as between glucose and serine. The bonding in nucleic acids is different. Here, the 3' carbon atoms of one sugar of a nucleotide are linked to the 5' carbon atoms of the sugar of the succeeding nucleotide by a phosphate moiety. The phosphate and hydroxyl groups of sugar join to form an ester bond. The ester bonds formed by the phosphate group between the 3rd and 5th carbon atoms of adjacent pentose sugar rings are called Phosphodiester bonds. This bond links 3' carbon atoms of one sugar molecule with the 5' carbon atoms of an adjacent sugar molecule. The phosphodiester bond is very
Final
vital to
structure.
all forms of life, as it
Double
forms the
helix
backbone
structure.
of deoxyribonucleic acid or DNA strands. According to the famous Watson-Crick model,
Watson-Crick,
the B
the
form of
great
DNA
scientist.
is the native conformation of DNA in solution. The B form of DNA is a metabolically stable configuration. Some of the features of bDNA are that it exists as a double helix. Two strands of polynucleotides run in opposite directions. In other words, they are antiparallel. That is, one strand runs in 3' to 5' and the other strand runs in 5' to 3' directions. The backbone of DNA is formed by a sugar-phosphate sugar chain. The nitrogen bases face inside and are almost perpendicular to the backbone. The 4 bases found in DNA are adenine, cytosine, guanine, and thymine, which are abbreviated as A, C, G, and T respectively. Each type of base on one strand forms a bond with just one type of base on the other strand. For example, adenine on one strand base pairs with thymine on the other strand by a double bond. Similarly, cytosine on one strand base pairs with guanine on the other strand by a triple bond. Each strand
Cytosine,
appears like a
guanine
helical
between
staircase,
triple bond.
and every step of
Adenine.
ascent is represented
Double
by a
bonds.
pair of bases. The 60-degree turn of the helical strand would involve 10 steps or 10 base pairs. In other words, there are 10 phosphate sugar groups in every full revolution of the DNA strand. The distance along the helix axis corresponding to one turn is known as pitch. In a B DNA, the pitch is 34 angstroms. The rise per base pair, or the distance between two base pairs, is 3.4 angstroms.
Yeah.
The different types of bonds we have studied are crucial as they provide structure and stability to biomolecules and make them functional.
This is the full studies about of biomolecules and their arrangements. Yeah, now is bacterial culture and growth, different types of bacteria. Yeah, this is one of our laboratory practice, bacterial growth and types of bacterial cultures and the system, how to the bacteria is develops. All the bacteria are not harmful one. Bacterial load through, we can enzyme, a lot of enzymes we segregate, enzyme segregations in artificially in laboratory conditions, erythromycin like medicines. Erythromycin medicine is through one of the bacterial segregation in the bacterial culture. So only we can procure. So you find it here. Bacterial growth, bacterial development. Okay. So listen the outlines of the bacterial, the culture types.
And what are the different phases of bacterial growth, everything. Now that growth is going to be studied in different types. So let's see what are those types and in detail about their uses also. So basically the
Four
bacterial growth
types of
systems or
bacterial
culture
systems.
systems are going to be classified into four types. How many types? Four types. What are they? Batch culture system, then continuous culture system, synchronous culture system, and biphasic culture system. So batch culture system, so it will be, the growth is going to be considered in a batch wise system. So those are going to be called as batch culture system. More oftenly we are using in the laboratory conditions. And then coming to the continuous culture system, which is very useful in the industrial purposes, where the culture is going to be continued. That's why the name is going to be the continuous. Synchronous culture means all the cells are going to be in the same size, so that is going to be the synchronous culture system. Then biphasic, where the two substrate molecules are there and the bacteria is going to show the two growth humps, so that is going to be called as biphasic culture system.
Okay.
So we will discuss
See the
all these four
differentiations.
culture
This particular
systems or
four types.
bacterial growth types
Four
in
culture
detail
systems.
now one by one. So let's begin with the first one, batch culture system. So a batch culture system is going to be of a system or a culture in which the medium is retained in a limited volume and is not supplied with fresh nutrients at any stage of growth. So we know that why we are taking this batch culture or continuous culture means because of the bacterial small size, we cannot study their growth by taking a single bacterial cell. So obviously what we will do, we will inoculate the bacteria in a liquid medium that liquid medium containing the little amount of the bacteria is going to be considered as a batch culture system. And a batch culture system is that system which involve the cultivation of a bacteria in closed culture vessel with a single batch of medium. So a batch, once it is going to be, if a medium is going to be inoculated with the bacterial culture and it was closed, and it have to take all the nutrients and it have to grow, and there only it have to die. So that means it is going to complete its life cycle in that liquid medium. So that's how that is going to become a batch system or batch culture system. So let's see that in detail now. So you may have this seen this graph in the microbial growth. We call this as a growth curve. For example, when a bacterium is inoculated into a flask containing fresh culture medium and incubated, okay, and what happens, culture medium and inoculated, and obviously what happens, it enters a rapid growth phase during which the bacterium divides by subsequent binary fission and increases its population in the flask medium. So that means you are going
Okay,
to
the
take a
growth
flask
system, one
medium
is
and you are inoculating
time and
a
number,
bacterium in it
the number of
containing a
bacteria's
very fresh
growth.
culture medium
So
and
to
incubated.
inoculate means it's
Sorry, here it is not
initiation of the growth.
inoculated, it is incubated. Now what happens
The
after
temperature of the
incubation? It is
incubation
going to
period and all
enter
these
into the
fine.
growth phase. So during which the bacterium divides by subsequent binary fission and increases the population in the flask medium. Since the bacterium are not transferred to a new medium or no fresh nutrients are added to the medium, so what happens? The increasing population of bacterial cells after a certain time enters into a phase called as stationary phase. And this entering of stationary phase is because of the exhaustion of the required nutrients and the accumulation of some inhibitory end products in the medium. And what happens eventually? The stationary phase of bacterial population accumulates into the death phase. So obviously, though still it is not going to be supplied from external environment like nutrients or the culture, or they are going to be transferred into another new media. So this stationary phase is going to lead into the death phase when the viable bacterial cells begin to die. So this is what happening in a batch culture system. That means they have to divide and they are going to die in the same culture medium. Suppose if we are going to take all these readings and collect the data and plot a graph, okay, and we are going to get the graph called as growth curve. So this typical growth curve is obtained by a batch culture system. So we are going to have a plot of this growth curve consisting of how many phases? 4 phases are there. What are they? Lag phase, log phase, stationary phase, and death or decline phase. So we had a detailed discussion in another part called as microbial growth. You can go through in it.
The
Okay. Now,
microbial
the
growth,
characteristic growth
these are the
curve
phases takes
obtained
place.
contain how many phases? 4 phases. So those are all the 4 phases here. Okay. Now, this is all about a batch culture system. That means we are going to take a bacterium that is going to be inoculated into a fresh culture medium and incubated, and it have to undergo all the phases, that is from the initial incubation level to the death phase in the same culture medium flask. So that completion of its life cycle or phases in that closed environment of the medium is going to be called as a batch culture system. Then what are the advantages of this batch culture? So the batch culture is very easy to set up because once you are going to take a limited volume of the medium and you're inoculating, and once it is done you are removing it. So it is easy to set up, and the environmental conditions are also easy to control. Then the type of vessel used can be used for different process of different times. For example, if you're taking a single conical flask now, then you can wash it and you can reuse it for other purpose. I'm talking about only conical flask. It may be for fermenters also. If the culture becomes contaminated, only one batch that is lost. Suppose any contamination occurred in the culture vessel, so you can throw out that batch only. No need of throwing all the batches. Okay. And the level of nutrients drop, which can create the conditions necessary for the microorganisms to manufacture secondary metabolites such as penicillin, is the best example of this batch culture system. So these are all the advantages of the batch culture system to grow the microorganisms. Okay, so this is all about the batch culture. That means once you are inoculated the bacterial culture in the medium, you are done with that batch. So that's how this is a batch culture system. Then what is the second type of growth type or the bacterial culture system is continuous culture system. Continuous culture system. So there will be the continuous growth of the microorganism. So that's why this is going to be named as continuous growth culture system. So it is a type of culture in which nutrients are supplied and end products are continuously removed. So for example, if you are going to consider this as a growth system or a growth chamber, so what happens, the microbes never reach the stage of exponential phase because the end products are continuously removed. So that in which the exponential growth phase of bacterial population can be maintained at a constant rate for a long period of time by continuous supply of the fresh medium from a reservoir. So this is a reservoir of the fresh medium to the growth chamber and continuously removing the excess volume of the culture medium of growth chamber through a symphony. So this is a tube symphony overflow. By doing so, what is the thing that we are going to get because of this means the microbes never reach the stationary phase because the end products do not accumulate because we are removing the spent medium, isn't it? So obviously what happens, the microbes are going to retain only in the exponential phase, and the continuous culture system can be operated in two methods. One is going to be called as chemostat method and turbidostat method. Okay, and we can discuss these chemostats and turbidostats now. So this is a chemostat, the continuous culture system by chemostat. So what is happening in this chemostat? So this chemostat is constructed so that the sterile medium is fed into the culture vessel at the same rate as the medium containing microorganisms is removed. Like that's the main step of this continuous, isn't it? So for example, if you are going to have a 10 flow rate, 10 ml of the flow rate, at the same time 10 ml of the spent medium culture is also going to be removed. Forced aeration. So here you are going to have a magnetic stirrer to supply. If it is a large chambers, there will be forced aeration and mechanical stirring of the cultures are used to ensure the optimal oxygen. For the supply of the oxygen, either the mechanical stirring is done or the forced aeration is going to be done such that the oxygen is going to be supplied equally throughout the vessel. Okay, so that's also very important. And the culture medium for a chemostat is on essential nutrients. Okay, for example, take amino acids or growth factors in the limiting nutrient. The growth rate is going to be determined by the rate at which the medium is fed into the culture vessel. So here we are going to maintain one of the nutrient as a growth limiting factor. Growth limiting factor. So that can be anything, either the amino acids or any growth factors. Now the rate of nutrient exchange is expressed on the rate called as dilution rate. More the dilution, high will be the microorganism. So if the dilution rate rises, so what happens? That means if you are having the dilution rate rises, the higher the microorganisms can be washed out of the culture vessel. So that should not happen. So you should have the low dilution rate such that both the cell density and the growth rate is going to be high. So it is inversely proportional to.
Chemostat.
If the dilution rate is more, so obviously microorganisms will be less. So you have to maintain the low dilution. That means it should have the more nutrients such that more microorganisms are going to be grown. So that's how at low dilution rate, both the cell density and growth rate is going to be high. If the dilution rate is high, it will be less. That is, microorganisms growth will be less. Why less? They are going to be washed off into the spent culture bottle. So this is all about the chemostat, where a chemical substance is going to regulate this continuous flow or continuous culture system, and that is going to be named as chemostat. Then moving to the second type, that is turbidostat. The second type of continuous culture system is turbidostat, and it has a photocell. So you can see here it is a photocell that measures the absorbance of turbidity of the culture in the growth vessel. So here the turbidity of the medium is going to act as a thing of recognizing whether the flow of medium have to be get into the vessel or not. Whereas in a chemostat, a nutrient, that a chemical nutrient is going to influence the rate of flow. Okay, the flow rate of medium through the vessel is automatically regulated to maintain a predetermined turbidity or a cell density. So this is going to be the light source and the photocell. If the density is going to be more, what happens? Automatically this valve is going to be opened and the medium will flow into it. If the density is going to be diluted, this will be closed and the medium is going to not flowing into this chamber, that is a growth chamber. Okay, so this is how the on and off light is going to regulate this photocell, which is indicating directly the turbidity in the growth chamber. So this can be comparable to our regular what we can it as a regulator. Okay, so the regulator shows us more current flowing or less current flowing. So the best example, check automatic what we call as iron box. If more heat is going to be inside, obviously the light will off.
Yeah.
If less heat is going to be there
Automatic
in the iron box, the light
iron
will
boxes.
on. So the same thing is happening here. Just it's an example to
Same.
understand I gave you. Okay, so here the flow rate of medium through the vessel is automatically regulated to maintain a predetermined turbidity or cell density. And the turbidostat differ from chemostat in several ways. The dilution rate in the turbidity varies rather than remaining constant, obviously, isn't it? The culture medium in a chemostat lacks a limiting nutrient, isn't it? But here the turbidostat is not having any limiting nutrient. There it is having. The turbidostat operates best at high dilution rate, whereas the chemostat is going to have at the low dilution rate, which is going to be of reverse to this chemostat, isn't it? So that's how you are going to have this turbidostat. So the second type of continuous culture system is the turbidostat. So we had gone through the two types of continuous growth system. One is the chemostat and the turbidostat, where the continuous flood of fresh medium into the growth chamber and continuous removal of the spent medium is going to be done. That's why this is going to be called as a continuous culture system. And then moving to the advantages of this continuous culture system is very used to study about generations of a single bacterial culture. In a batch culture system, we can't do that one, but we can see only one generation. But here in the continuous culture system, we can study about many generations of the single bacterial type or bacterial culture. And this system, that is a continuous culture system, make possible to study the microbial growth at very low nutrients level. Okay, and this is also maintained in a constant environment for longer duration time in a continuous growth culture. And especially this culture system is used in genetical and biochemical studies where there is a necessary to maintain prolonged exponential growth of bacterial population. And this type, that is this culture system, is also very beneficial in industrial process. So these are all the some of the advantages of this continuous culture system. Then moving on to the third type of the culture system, that is synchronous growth or culture system. The synchronous growth of a bacterial population is that during which all bacterial cells of the population are physiologically identical and in the same stage of cell division. That means if you are going to take a class, LKG, nursery or UKG, they're all going to be of the same height. Okay, exceptions are there, but basically they are going to be of a equal in size. We can't compare a LKG student with a 10th class, isn't it? So that's how, depending if all the cells are going to have the physiological identical and in the same stage of cell division cycle at a given time, that type of the growth is going to be called as synchronous growth. Excuse me. Then what is the significance of this synchronous growth? Synchronous growth helps studying the different stages of cell division cycle and their interrelations. And one more thing that we have to remember here is a synchronous culture can be obtained either by manipulating environmental conditions such as repeatedly changing the temperature or by adding fresh nutrients to the culture as soon as they enter the stationary phase, or by physical separation of cell by the centrifugation or by filtration. See, this synchronous growth in most of the bacterial cultures, the stages of the growth and cell division cycle are completely random, and thus it becomes difficult to understand the properties of these bacteria during the course of division cycle using such cultures. What cultures? That is continuous cultures and our batch cultures. To overcome that one, we are going to opt this type of the culture called as synchronous growth or synchronous culture system, where all the bacterial cells of the population are physiologically identical and in the same stage of cell division cycle at a given time. This is very important. At a given time, all the bacterial cells are going to be physiologically identical and in the same stage of cell division. Okay, so as I told, we can get this synchronous culture by manipulating environmental conditions or by changing the like temperature or by adding fresh nutrients and not allowing them to get into the stationary phase, or by physical separation or by centrifugation or by filtration. So these are all the different environmental conditions. If you are manipulating, we may get this synchronous culture or growth of bacteria. So the most widely used method to obtain this synchronous culture is the Helmstetter-Cumming technique. Helmstetter-Cumming technique. Okay, so here is the one. And according to him, in this Helmstetter-Cumming technique, an unsynchronized bacterial culture is filtered. So here is the unsynchronized filter, that is bacterial culture, is filtered through a cellulose nitrate membrane filter. So where it is going to trap the thing. It's like a sieve, okay? The loosely bound bacterial cells are washed away from the filter. So it is going to be removed, okay? And the leaving some cells tightly associated with the filter. So some of the cells are going to be trapped in between the pores of this cellulose nitrate filter, and some of them are left behind and some are washed off. Okay, now the filter, now we have to take this filter, that is a cellulose nitrate filter, and we are supposed to invert it. So you are going to invert it. And now you are going to pass a fresh medium to flow through it. So you have passed a cell culture, okay, unsynchronized, that means different sizes of the bacterial cells are there, and that culture was passed through the cellulose nitrate membrane filter. Now what happens? Some were washed off and some were left behind and some were trapped in the cellulose nitrate membrane. Now you have to take that cellulose nitrate filter, that is nitrate membrane, and you have to invert it, and now you are supposed to pass a new fresh medium, through this nitrate filter. Now what happens? The filter is now inverted and the fresh medium is allowed to flow through it. Now the new bacterial cells that are produced by cell division and are not tightly associated with the filter are washed into the effluent, isn't it? Into the effluent. Hence, all cells in the effluent are newly formed and are therefore at the same stage of growth and the division cycle. Thus, the effluent that we are getting represents a synchronous culture or synchronous growth. Now, did you understand what I said? So, here are the different sizes of the bacterial cell. And when you pass them through a filter, that is a cellulose nitrate membrane, some of having the big size will left behind and very having the small size are going to be coming with the filter, and only the equal size of the pore size of this membrane are going to be trapped in that membrane. And now you are going to take that membrane and kept in an inverted position in another chamber and passed, uh, allowed to flow through a fresh medium. And now what happens as the fresh medium is passing through this filtrate, these all cells tries to have the cell division, isn't it? Now once they are having the cell division, because of all of them are of the same size, now all of them get start of cell division. They will be of the what we call equal size and they are of at the same stage of cell division. Now when you these all small size particles or bacterial cells will pass through the effluent. That means they come along with the effluent, and if you observe that effluent, then we can find all the cells are of having the same stage of growth and division cycle, so which we call it as representing as synchronous culture or synchronous growth. So this is all about the Helmstetter Cumming technique to explain about the synchronous culture system. Then moving to the last type of the growth, that is biphasic growth, or it is also called as diauxic growth. What is called it as diauxic, AUXIC, diauxic growth. So this biphasic or diauxic growth is represented by two growths. So if you see here, we are having the two growth curves intertwined by a short lag phase. So here basically we'll have a big lag phase, but here we are going to have a very short lag phase produced by an organism utilizing two different substrates. One can be a glucose and other can be something. So here, if you observe a particular type of the growth curve seen in a cultured organism in which they have two exponential phases. This is first phase of exponential and this is the second phase of exponential growth. And those two are separated by a small plateau phase, which we call it as a shortened lag phase. And this double hump curve is produced when it is going to be produced. Two types of growth curve is produced when the microbes are cultured during two carbon sources, one of which must be used up before the second can be used. So you are going to have the two types of things to eat. So whatever you are going to like first, that you'll eat, and other one you will going to take it when you are feeling hungry. Okay, so in the same manner the bacteria is also doing the same thing. So when an E. coli grows in a medium containing both glucose and lactose, what it will do, obviously, it will take the glucose first, preferentially it will utilize the glucose first. And we are going to have a growth curve like this. So when the glucose is going to be exhausted, then what happens? So it will slightly think of what to do. Shall I eat the thing that I don't like? So obviously it will start up eating the lactose and starts up using the lactose. So slowly again it grows in size, and this is the second phase of exponential source. If this diphasic growth of E. coli is going to be plotted in respect to bacterial density and the time taken, you are going to get this two growth curves follow one after the other with a short phase to produce a diauxic or biphasic growth curve. Okay, so by this we are going to have an idea that biphasic or dioxic growth is nothing but one where you are going to see the two growth curves, and in that two growth curves we are having the two lag phases intervened with a short lag phase, and this is going to be observed because of having a choice of two carbon sources. The first carbon source utilization, you will get one growth curve, and the second carbon source, it is not going to enter into the stationary phase until it is going to utilize the second carbon source. So here you can see now the bacteria has entered into the stationary phase because we do not have the other option of the third carbon source, isn't it? Obviously, it will get into the stationary phase. So this is all about the last type of growth system, that is dioxic or biphasic growth system. So thus we
Okay.
end our growth systems, that is bacterial culture
Or
system
growth
or
systems.
bacterial growth systems. Thank you.
Okay. Okay, this is of our bacterial growth system, the laboratory conditions, how to bacterial growth is possible. Okay. For bacterial growth, what apparatus is needful? One is laminar flow, petri dish, conical flask, the media.
Prokaryotic cell
Prokaryotic is
reproduces by
bacteria.
dividing, creating two identical cells. This process is called cell division or binary fission. The rate of replication varies between different types of bacterial cells. E. coli cells are preferred for laboratory experiments
Laboratory
because they
experiment E. coli
divide in a matter
cells.
of minutes. In nature, bacterial cells grow in a variety of settings. In a laboratory, bacterial cells are typically grown either on a solid medium called agar or in liquid broth. Bacterial cells that replicate on a solid surface pile on top of other cells, forming a visible spot or colony. Bacterial cells that replicate in liquid culture will
Yeah,
float,
this is
giving the
shaker.
culture a cloudy or
Shaker
turbid
shakes
appearance.
towards.
For cell reproduction to occur, cells need to have the right nutrients: acidity, temperature,
These influencing
oxygen,
factors
and moisture.
is requires.
These conditions are easily controlled in a lab, making bacteria a popular choice for large-scale protein production. For example, E. coli are used to produce large amounts of insulin for diabetic patients.
insulin
Bacterial population growth occurs in
secretion.
4 stages. Progression through these stages is regulated by the availability of space and nutrients in the environment. The first stage is known as the lag phase,
These all
because
phases on
although
one
the
culture
cells are
we are
dividing
identified.
quickly, the number of cells is
The
still
lag phase,
small.
expansion
If an
phase,
initial E. coli cell culture
stationary
started
phase,
with
and
one
death
cell,
phase.
after 2.5 hours
The
of
death
growth,
phase we
there would be
extract.
more than 500 cells. Growth during the lag phase is slow because of the low number of bacterial cells replicating and the time needed for bacteria to synthesize the proteins needed